Okay, so here what we're going to do is we're going to look at enzymes, what they are, how they work, and all that good stuff. So, um, let's start. Now, enzymes are biological catalysts, okay? Enzymes are biological catalysts. So what this means is that they are made from DNA. That's how they are biological, okay? So they're coded for by DNA. And like we know, anything that's what DNA codes for is proteins. So enzymes are specific proteins shaped in a particular way. And so that's what they are. What they do is they speed up the rate of reactions, speed up the rate of reactions, which is brilliant. And they themselves are not used up, so they are not used up. So if you have 5 grams of enzymes to begin with, you're going to have 5 grams of enzymes at the end of the reaction. They don't get used up. All they do is they speed up the rate of reaction. So the question is, where might they be used? Number one, in respiration. Okay, you need enzymes in respiration. You need enzymes for photosynthesis. You need enzymes for digestion. Now guys, I'm going to tell you about this digestion one. Okay, now with digestion, I've already covered this in a previous video where I've done the exam paper, so you need to know what the specific enzymes are. You need to check out that video where I did the November 2012 exam papers. Check the video descriptions and that'll give you a better idea of where exactly I covered digestion, but you need to know the different enzymes involved. Now number 4, what you don't know probably, well you would do if you're higher because I've already covered this for higher material, but you need to know this for foundation as well, is that they are involved in unzipping the DNA. Okay, so as you already know, A and T and G and C are held by weak hydrogen bonds. Okay, A and T are held by 2 hydrogen bonds and G and C has 3 hydrogen bonds. So an enzyme will come and unzip the double helix DNA so that strands of DNA are exposed. Okay, so that's another thing that enzymes do. So all of these here occur within an organism. So respiration within all organisms, photosynthesis within a plant cell, digestion with animals happens within the body, and unzipping DNA happens within a cell. However, not all enzymes necessarily work within the within a body or within an organism. Okay, so for example, what we've got here is we've got fungus. Now you should all know from B1 that fungi are saprophytic feeders. Saprophytic feeders. Okay, and so what this means is that they release enzymes into the environment, and once they release it into the environment, the enzymes end up breaking down the food, and then the fungus absorbs the broken down food. So that's just to show you that it doesn't have to be within a cell. Enzymes, biological enzymes, can also work outside of cells too, in the case of fungus. So now next what we're going to do is we're going to have a look at how enzymes actually work, and that's what we're going to do next. So the question is, how do enzymes actually work? And the answer to this is called the lock and key hypothesis. Okay, so that's one of the keywords that you need to be aware of, the lock and key hypothesis. So what this says is that every single enzyme has a specifically shaped active site. So this is my active site. Right, and with an active site, only specific substrates can fit in. So if I've got these substrates, these 3 substrates, question is, which one is going to fit that active site? Okay, and the answer is, of course, it's not this one because the shape is wrong. It's not this one because the shape is wrong. It's this one here. Now, there's specific wording that you need to know about this. So this substrate has a complementary shape to the active site of the enzyme. So it has a complementary shape to the active site of the enzyme. You can't say it's the same because it's not this. If it was the same, it'd look exactly like this. It's not the same. It doesn't look anything like that. It's complementary in shape, so it can fit into that active site there. So that's what the lock and key hypothesis is, that every enzyme will have a specifically shaped active site, and only specific substrate with a complementary shape can fit into the active site of an enzyme. So that's lock and key hypothesis done. Next, what we need to look at is how enzymes behave and how they work under specific circumstances. Okay, so we're gonna go with pH first. So if I've got a graph here of enzyme activity against pH, so here's my pH scale, here's my enzyme activity, I'm just going to write EA for short. Now I'm going to look at one that I've covered in the digestion video already. Again, look back at that video, it's when I did the exam paper, it's in there somewhere. So the enzyme within the stomach is called pepsin. Okay, now pepsin belongs to a family of enzymes called proteases or protease. Right, and now what proteases do is they break down proteins. So they break down proteins into amino acids. Now, with this, that's what it does. Breaks down proteins into amino acids. Now, because it's found in the stomach, right? The stomach has a pH of about 1 to 2, right? Let's settle with 2. So it's about a pH of 2. So this enzyme needs to be able to work under this acidic condition. So if I put the pH scale here, 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, it'll go up to 14. That's what alkali is. This is pH 7, which is neutral. This is all acidic, up to 6. So its maximum activity, the enzyme's maximum activity will be at pH 2 because that's what it's adapted to deal with. So I'm going to put a cross there. Next, what you're going to find is that the activity is the highest at this point, and then it goes lower at this point. The question is why though? And the answer for both this part here and this part here is the same. It's because the enzyme becomes denatured. So what does that mean? Basically what that means is that the active site of the enzyme changes shape. And so why is this a problem? Because if the active site changes shape, then the substrates can no longer fit. They can no longer fit the active site, and so the enzyme activity is going to decrease. So here it's too acidic, and here it becomes to get less acidic, more towards neutral and alkali. So the enzyme is going to denature. Now what you gotta remember is that different enzymes will behave under different conditions. Okay, so there are some enzymes that will have, for example, they will work best in pH 7. So the graph for that enzyme would look something like this, where the activity is lower, reaching a maximum at pH 7, and then decreasing again. Something like, okay, so this it's a bit dodgy line, but whatever. Again, the reason is the same. It gets denatured here, maximum activity here. And you could do the same for any enzyme under any pH. So long as you know what the best pH is for it, you just draw the maximum there, and on each side denaturing happens, i.e. the active site changes shape, substrate no longer fits. So next what we're going to be looking at is a graph of temperature against enzyme activity. So here's my enzyme activity, here's my temperature. Now within the human body has a temperature of 37 degrees. So in other words, that's where the maximum activity will happen. So I'm gonna have my maximum activity here, and here we're gonna have low activity until it reaches the peak, and then it's gonna drop off again. Now the reasons for both sides is different though. Where it's a low temperature, so here the temperature is lower, there's not enough energy. So there's not enough energy, which means that the rate of reaction is going to be slow. So rate of reaction is equal to slow. It's going to be slow. Now with this bit here, what's happening is it's getting too hot. So there's too much kinetic energy. Which means that the active site will change shape. So it's going to change shape, and the substrate no longer fits. The substrate won't fit the active site. And what is this called, guys and girls? That's correct. This is denaturing. The enzyme at this high temperature ends up becoming denatured, which means that the active site will change shape and the substrate won't fit anymore. That's what happens when it gets too hot. That's why the enzyme activity is decreasing here. So next, what we've got to look at, and this is the final bit for enzyme, is I dropped my pen. Next, what's going to happen and what we're going to be looking at is the following. It's going to be enzyme activity with substrate concentration. Okay, so here's my substrate concentration, and that's the shortcut line sign for concentration, and here's my enzyme activity. So what happens first is that as the reaction begins, the active sites begin to fill up, okay, and all the substrates are going into the active sites. Then what happens is it begins to level off. Why? Because all of the active sites are filled with substrates, okay, and so it's kind of leveling off. The activity is leveling off. So hopefully that's explained that. So this part here, the active sites are being filled with substrates. Here all the active sites are filled with the substrates, and that's that. So the enzyme activity kind of levels off, and to increase this you're gonna have to introduce more enzymes. Okay, that's the only way to increase it really. And that is the 3 graphs that you need to be aware of, and that concludes our video on enzymes. And so that means I'm gonna have to upload this as well.